everyone, and welcome to the Kids First Winter webinar celebrating 10 years of Milestones and Momentum. I'm Vanessa Barnes. I'm with the Office of Strategic Coordination at the Office of the Director, and I'm a public health analyst. I'd like to quickly show our agenda and get into some housekeeping. We have a stellar lineup of panelists and speakers, and I'm so excited for you all to hear what they have to say. I'd first like to welcome Dr. Vivian Oda-Wang, the Deputy Director of the Office of Strategic Coordination, to give our welcome today. Dr. Wang has an extensive career in genomics, science policy, and data governance. Dr. Oda-Wang served as a thought leader in the early stages of Kids First and provided her expertise on key data issues. Dr. Oda-Wang, welcome.
Thank you so much, Vanessa. Have to say, it's so nice being back with you after all these years. So as you may remember, I helped establish and chaired your Data Access Committee years ago. So I'm Vivian Oda-Wang. I'm now the Acting Director of the Office of Strategic Coordination, better known as the Common Fund, where you're sitting here at NIH. The Common Fund was pleased to implement the first 10 years of your program. And today, for me, it is really wonderful that we can continue to administer your program for the next 5 years on behalf of the NIH Director. I'm especially appreciative of the opportunity to thank you for your tireless advocacy for the Gabriella Miller Kids First program. Together, we have created one of the largest and most comprehensive open access pediatric genomic resources in the world, with data from children affected by childhood cancer or congenital anomalies and their families. Importantly, we have enabled researchers to uncover shared genetic mechanisms. And by making this data easier to share, we've been able to really just upgrade research and accelerating discoveries on behalf of all children. So in November, Gabriella Miller Kids First released its newest study, as you know, Kids First Extracranial Germ Cell Tumor Study. This new data set includes information from 393 children and young adults, along with their biological samples. Researchers now can access the genetic data from these participants, and in many cases, their parents. Data on genetic changes found specifically in tumors, and information that shows genes that are active in the tumor. This study nicely complements existing Kids First data sets on intracranial germ cell tumors, providing researchers with a more comprehensive view of how these cancers develop both within and outside the brain. I'm just so happy that today we're celebrating our achievements and talk about what we're going to do going forward. I can't think of a better guide to start this discussion than my longtime colleague, Larry Brody. Although you may know Larry as a steadfast champion of the program since he was involved at the beginning, he's also he's a bit too humble, but he's also a stalwart and beacon in the field of genetics and genomics. His day job includes the research bench, where he has a lab and is a senior investigator of the Social and Behavioral Research Branch, and leadership as the Director of Genomics and Society at the NIH's National Human Genome Research Institute. But as you know, as I know, he is a force and really at the true forefront of ensuring applying genetic information and addressing its ethical implications to research, clinical care, and public health. Larry, what a treat to see you today. I'm turning it over to you.
Thank you, Vivian. I'm blushing a bit. I'm here today to talk about Kids First, which, as Vivian mentioned, I was there at the birth when, after Mrs. Miller and Congress agreed to create a program that was focused not within an institute, but across institutes. And so my task today is to give you a very, very short introduction to the last 10 years. And we're going to spend most of our time in the future, which is where we really ought to be. But I will say that it's been really gratifying to see a project go from a nascent idea and some discussions between the NIH director and the director of NCI more than 10 years ago to a fully shaped program that is really having a big influence on the field. So I have a few slides to show you to set the scene for those of you who have not been following Kids First for the last decade or so. So I'm going to share my screen. So let me just cover the priorities of this program, which really were there from the outset. A lot of research programs evolve over time. This one was very, very set in nice foundational principles from the beginning. And so program priorities included a user experience, meaning that from day one, the idea that this program would widely share data. And I will come back to the data sharing thing again and again in this brief presentation. There was also that NIH would have governance, so it would be an NIH program, even though almost all the hard work was being done by people in the outside of the NIH. It was an early adopter of cloud computing, which was a buzzword for a long time, but Kids First helped make it a reality. And then the data would be set up to be interoperable with lots of other data sets in the NIH ecosystem. And as I already mentioned, the sharing would be a major tenet. I'll come back to the interoperability because that's one of the things that we still need to work on and we are working on. There's been tremendous success in the other areas. First, I'll just highlight the data portal, which you'll hear more from Adam in a few minutes. There is a tremendous amount of data available to researchers and a certain level of it to the general public from 30,000 patients. And I've been in genetics for a very long time, more than 40 years now. And it's amazing to just say that we have genomes from 30,000, whole genomes from 30,000 patients. And that the data is not good unless it's being used. And you can see here on this slide, approved data access request, which I believe will continue to accelerate, over 1,000 approved sets. And this is from people all over the research spectrum. The diagram on the right just shows you some of the systems that are covered by these data sets. And I'll just make a note, I'm pretty sure these slides will be available if you don't have time to read all to see whether or not your favorite system is covered in the diagram. This slide just talks about the increasing impact, and any project that starts from zero takes a while to ramp up, but you'll notice that we have been in the steep part of the curve for the last 5 years. I would expect that the publication citing will see another inflection point, and the publications will even accelerate as the data increases. To give you an idea of some of how the data are used, I'm not going to go through each of these publications on these slides, but you can see that the data are being used by researchers all over the place who didn't create the data but are using it. And that's the idea of this program, is to spread the data and get as many users as possible to take advantage of this investment. And so it includes on this slide are a couple of examples from the cancer field. There are also birth defects uses as well. There was a connection that was made at the beginning of this program between birth defects and childhood cancer, because the program is doing both. This was not widely appreciated that there could be a big connection at the outset of the program. And as many of you know, NIH has different institutes, one that would study birth defects and one that studied cancer. And one of the major successes of this program is the bringing together of these two efforts and the convergence. And you'll see here's a few publications on this slide that are connecting birth defects and cancer. If the program had been cancer alone or birth defects alone, we wouldn't have had this convergence. And now investigators who are studying cancer and birth defects somewhat rendezvous at the Kids First data resource. I just wanted to close with a few elements of looking forward. This is a program, as some of you may know, my institute, the Genome Institute, has pioneered data sharing and spread to the entire NIH. And this is a real paradigm shift and an amazing use of the investment of the taxpayers' money to generate data, to be able to share it with both researchers, and the participants. And as Vivian mentioned, she's been at the forefront of data sharing And this idea of sharing data with participants turns out to be a really good idea that some people resisted at first. The other reason that data sharing is so important is it really democratizes the data, so that it may be that the data is generated in a very high-resourced environment. Anyone with a good idea, no matter where they are, can test their idea on this data, and they don't have to be in a high-resourced environment. They just need good ideas, which don't necessarily track with geography all the time. We're looking forward to expanding conditions to be included. There is still more that could be done. And then, as I just said a few minutes ago, that the highlight is this merging of conditions that would have been siloed if they had been done separately by different institutes. And we're hopeful that the Congress will continue to support this program for many years, because as the data gets data set gets larger and larger, the value only goes up. I'd just close here with thanking the folks. This is a really big effort, and one of my real joys at working at the NIH is working between institutes, between disciplines, and this program has the hallmark of it. It's managed by the Common Fund. It's staffed by lots of different NIH staff. It works across, I should have made a slide of all the institutes and centers that participate in the program, but there are many. There's a steering committee that is from many different institutes that I'm on the steering committee. But we also really have a lot of gratitude to the researchers, some of whom you'll hear from in a few minutes. And I can't forget the families and the participants, because there'd be no data without their willingness to participate in this study. I just want to close with a nod to Diana Bianchi, who is no longer at the NIH for reasons that are completely unrelated to Kids First. She was a stellar supporter of this program, and I just want to honor her by conveying my gratitude to Diana. And with that, I will turn it over to, you'll hear a short video, and then we'll hear from Adam Resnick. And I will be around for the duration of today, so I'm happy to answer questions if you have them.
It's gonna be okay.
I don't know what's gonna happen.
Well, first, we're going to get ice cream.
Ice cream?
Thought you might like that idea.
I'm gonna get chocolate with extra sprinkles.
Before you know it, the first day of school. And your birthday is coming up.
Yeah, wishful thinking.
You still have so much growing up to do.
Happy birthday to you.
And I know it won't always be easy. But I know you're gonna do great things. You'll build a life that's all your own.
I can't believe I'm gonna have a baby. Oh my god, I'm gonna be a grandmother.
And I can't wait to see everything that happens. Sarah, we're ready for you. Okay, you ready? All right, let's do it.
Wow, I'm so happy we got to share that video today. That video really underscores the importance of the Kids First program and the data's impact and future impact. Next, I'd like to present Adam Resnick, who is the co-executive director at the Center for Data-Driven Discovery in Biomedicine, also known as D3B, at the Children's Hospital of Philadelphia. Dr. Resnick provides leadership at the Data Resource Center, researches tumor progression in brain tumors, and also provides training to those interested in using the Kids First portal. Welcome, Dr. Resnick.
Thanks so much. Hopefully you can see my slides okay. And thank you to the previous speakers for really setting the stage. For us in the Data Resource Center, the 10-year mark really is a moment of reflection on the vision that the Kids First program began with, and really reflecting on both what we have achieved and also vetting whether the hypothesis we began the program with have been born. And I think that's one of the hallmarks of success in some ways for us, which is when things that didn't exist before or ways of doing things weren't being done before become a new norm or a new paradigm that we then take for granted. For us within the Data Resource Center, that is a true measure of success. But I thought I could just go through a few slides to sort of highlight that transition in ways that set the stage for the next speakers and the panel. And I think that's one of the really exciting parts about being part of the Kids First program and the Data Resource Center is that our job within the program is not just what additional data or what additional conditions or diseases require additional data and how to enable it, but it's really also the how. How can we transform the way science is done in conditions that are often rare, where no single institution might have enough patients to support the full research requirements of a cohort, or where no single disease or condition can develop the entire architecture and infrastructure required to ultimately advance a data-driven approach that ultimately drives a precision medicine or personalized medicine approach that is by and large a requirement for many of these conditions. And so when we first began the program, we really aligned into this hypothesis framing that if we could bring together a condition framework that spanned highly annotated samples and participants, generate large-scale modalities of data, particularly focusing on genomics, that through the integration and provisioning of that data across diseases and conditions, we'd create new information that could then drive new knowledge creation and ultimately impact for patients. And importantly, in considering the architecture and design of this new program, particularly from the perspective of the Data Resource Center, our goal was to ensure that we build a sustainable framework that accelerates and drives that process longer term through really leading edge platforms that, as Larry mentioned, harness new technologies at the time and aligned with the capacity to transform samples and data into knowledge and impact through community-based efforts that accelerate that process. And when we began diving deep into that framework and design principles, we outlined a few key design principles that I thought I'd reshare just so we can reflect at the 10-year mark on if and how we've been able to achieve those and what's left for us to continue to advance. From a platform perspective that addresses that unmet need of multimodal data across institutions, across investigators, across diseases, the goal was to develop a discovery knowledge creation framework that is non-local, meaning, as Larry mentioned, no matter who or where you were, if you had a good idea, you can drive an information-driven and collaborative global process for discovery. And the goal of the platform should actually be to maximize the number of information users and improve their secure access to the data and information. And that's important because these are really complex conditions and diseases that besides often what is a dearth of data and available data sets, there's also a dearth of investigators, not enough scientists dedicated to a particular disease context. These are complex frameworks. And so the paradigm we wanted to enable is to bring new investigators who may not necessarily have defined their domain of interest as disease X to come over from disease Y and intersect with disease X to empower that framework. And then the platform and ecosystem must support and maximize the diversity of information users with varied interests, specialties, and expertise on behalf of what we termed at the time convergence research processes. So this was clear that we needed clinicians who had deep expertise in the clinical framework of the disease to work alongside with genomicists and bioinformaticians, to work alongside with imaging scientists, to work alongside with computational researchers in ways that is by and large unprecedented and is incredibly complex, because each community in each domain requires its own set of frameworks of accessing, using, and integrating knowledge. And that if we're successful, we support our goal is to maximize the connectivity of the community, because connection and interoperability, as Larry highlighted, is the key for driving that maximized diversity and information user context. And so the platform must support continuous and ongoing connections, not just across the Kids First boundary, but to other resources, including for us, NCI and the Childhood Cancer Data Initiative frameworks, Biodata Catalyst at NHLBI, Terra and framework from NHGRI, just to name a few, as well as all other Common Fund programs under the umbrella of what's called the Common Fund Data Ecosystem. And those have been incredibly fruitful from an architecture of defining a new way of doing data-driven research. And importantly, as Larry mentioned, the key is to leverage these new technologies in ways that prioritize the frictionless, what we call the frictionless flow of data and information via trusted connections, data federation, and user communities. And reflecting backwards across the 10 years, for us in the Data Resource Center, it's very fulfilling to see that we have met many of these goals, but also recognize that there are new ways, new technologies, and still gaps left in that process in ways that we look forward to advancing in the coming years. Highlighting that success, I thought I'd just bring in a few vignettes of looking at that trajectory across time, beginning with, for me at least, a very salient context. My foundational work really began in pediatric brain tumors. And so for me, the Kids First Data Resource and its link to Gabriella was such a foundational metric of impact for the program. And so when Gabriella wisely guided us and the rest of the community to stop talking and start doing in ways that really was the harbinger and birth of the Kids First program. At that time, to my knowledge, there were no genomic data sets available for the disease that she suffered from, diffuse intrinsic pontine glioma. And that meant that any researcher who was going to try and bring to bear new knowledge on behalf of impact was limited to whatever they could find or garner on their own. Today, if you went into the Kids First portal and you search for diffuse midline glioma, you'll find that since that time, we've been able to actually subdivide and further classify gliomas, but you will find that there are more than 12,629 data files available for immediate use on behalf of diffuse intrinsic pontine glioma, again, a rare subtype of brain tumors that simply from a perspective of numbers demonstrate the immediate value proposition of the Kids First program. But importantly, that diffuse midline glioma cohort is contextualized across all other pediatric cancers and congenital anomalies in ways that the community can cross-query, cross-understand, and cross-interpret. And paired with those, genomes are imaging files and increasingly multiple forms of modalities, which I'll talk about briefly. That diffuse intrinsic pontine glioma cohort is part of an increasingly growing and expanding framework of studies that we're marching through releasing as a Kids First Data Resource, with 35 studies already on a portal, but 86 total studies provisioned, representing more than 53,000 patients and 9,000 family members, to my knowledge, the largest integrated platform across pediatric cancers and structural anomalies for mankind. And importantly, as Larry mentioned, building that architecture takes time, not just from a technology perspective, but also from a community perspective. And that technology and community perspective intersects as more than 1.56 million total data files are now available within the platform in ways that has scaled and grown over time, in part because the process of data generation has scaled and grown over time. And again, that's a key principle that we believed very early on in that is coming to bear, specifically that the number and amount of data we can generate scales as well as the number of users across time, creating this synergy interplay between that context that drive these curves of steepness across conditions in a unified way that span more than 2,500 conditions within the portal. The amount and type of data is one metric that we're very proud of. But as Larry mentioned, it's also the use and how the data gets used by the community. Here you can just see the number of portal users growing over time. But within the Kids First data resource, there's also a number of different ways of accessing and using the data. And another metric besides portal utilization is the approved and validated access to data across time. For those of us who look at charts all day long, whether it's in the scientific domain or in reporting metrics frameworks, when we see curves like this, the first thing I look at is the slope at the curve at different time, with the slope really representing change or acceleration over time. And so you can see really in the early days of the Kids First program, as Larry highlighted, a fairly flat curve as we were just beginning to march through the availability and amount of data that's available. But at every point, you can start to see that inflection take place. And the steeper the angle of the curve, the more rapidly things are changing. And across time, you can see that steepness of the curve is increasing, really highlighting that acceleration and synergy that brings together the amount of data, the amount of users as enabled by a platform. And for us in the Data Resource Center, this is one of the truest metrics of success because acceleration and time is the variable for impact that we are trying to drive on behalf of the patients represented by the cohorts. That acceleration and use of data happens because we are meeting that additional metric of expanding the scope of who it is that is doing the research. Historically, there was a very tight coupling from those who generated the data to those who made the discoveries. And we need to maintain and drive and empower those users who essentially partner with patient communities to collect samples, to enroll them on observational research protocols or clinical trials, and translate those samples into data sets that they can then further interrogate and drive new knowledge. And so here, that group are what are called the EXO-1 investigators. These are the talented researchers who really drive the curation and assembly of what are often rare disease cohorts across network, but are leveraging the Kids First program to advance new data generation on behalf of that analysis. And those are really the preeminent researchers in their field. Some of them will have a chance to talk on the panel shortly, driving new knowledge creation as enabled by the platform. But a second metric, that again, as part of our hypothesis framework and methodology, was to bring additional secondary users into that framework, meaning users who begin to come and leverage the data after it has been generated and released. And here's where you see that blue secondary user group growing over time, harnessing those cohorts that are being released on the portal and platform. And that user group of expanded secondary users also expands the scope of what they are studying, specifically blurring the boundary between disease or context of condition. And so here you can see the publications by domain as they have emerged. Obviously, you have publications on specific pediatric cancer conditions or anomalies, but that growing purple part of the pie that looks at cross-condition analysis is really a phenomenal metric of the success of the program. It's against that backdrop of putting together a platform-based approach that supports interoperability, multimodal data across domains and modalities, that we within the Data Resource Center are extremely excited for what's to come next and is already underway. Foundationally, within the Kids First program, generating the whole genome view of patients along with the transcriptome for tumors was a foundational layer that defined that first benchmark of a data-driven approach to interrogating the disease or condition context. But increasingly and excitingly, the capacity to drive new types of measurements at scale has emerged over the past few years in ways that the Kids First program has begun to support through either pilot initiatives or now ongoing initiatives. This includes generating some of the largest ongoing cohorts of long-read sequencing, a new technology that just emerged over the last few years that is driving deeper penetration and understanding of the genomic context for these patients, as well as proteomic and epigenetic and immunogenomic analysis on especially cancer cohorts that drive deep multimodal analysis of those tumors. And those are just emerging cohorts. Some of it, some of the speakers following me have already began to advance, but creating unprecedented increased knowledge frameworks that is poised for clinical translation. The Kids First program really began by harnessing that network of community stakeholders that connect further into the patient population framework for creating cohorts of samples that can be linked to initial phenotypic data. But what has also emerged over time is new ways of connecting that data and opportunities we look forward to harnessing in the coming years. Specifically, as we began to build that architecture that supports the integration of that deep omic data layer with associated phenotypes, we began investigating again as a community-based efforts across NIH, the implementation of tools and frameworks that supported deeper and potentially more rapid and ongoing integration of clinical data from clinical networks in ways that is poised to further advance how this data comes to bear on impact and how data continues to flow across time, so that a data set that is being provisioned within the Kids First data resource is not just a point-in-time data, but continues to advance as more data gets collected and assimilated about those patients, about their treatments and their outcomes across time in an ongoing, dynamic, and longitudinal way. New infrastructure that supports that dynamic view of patients across times with their samples does not yet exist, to our knowledge anywhere, in ways that we hope to continue to advance and build. But it comes at a time when, just as we are now enabling that non-local cloud secure environments of data integration across networks, we're now facing the opportunity of a new modality of analysis with the emergence of AI technologies. And it's that combination of complex, multimodal, multi-omic, multidisciplinary data in common environments that bring together those user groups and across space and time, newly enabled by emerging technologies such as AI-based frameworks of interpretation that really stage the vision for the next wave of transformational impact we hope to advance within the Kids First data resource in ways that we and many others in our community are eager to continue partnering with across this amazing, complex, and collaborative framework of innovation in ways that not only drive continuous discovery through publications and additional data resources, but also further accelerate how those come back into the utility of those resources in the day-to-day clinical domain of impact for patients. Achieving that will be the full success metric on behalf of that pithy requirement that Gabriella first enunciated at the origin of Kids First, of taking action and doing what's required on behalf of true innovation, not just on behalf of pediatric cancer and structural birth defect, but on behalf of how science is done altogether. And that could not have happened if it wasn't for that community engagement, continuous support, shared vision that both Ellen herself has continued to provide, that has broadened across a much larger community of parents, foundations, and patients and participants writ large, not just within the Kids First program, but in sister programs and efforts like the CCDI and Include program, as an example, in ways that I think, again, reshapes the how of science by fully integrating a community across patients and foundations and research in a unified space. So it's really through collaboration that, reflecting back, we have been able to drive innovation, and we look forward from the DRC perspective to that ongoing development. None of this is a single entity work, including the DRC itself, which spans a number of different institutions from St. Justine, Chicago, CHOP, and others, partnership with NIH across the institutes and centers, the Common Fund. It's taken really a true expanding village to achieve the first 10 years in ways that I think exceeded most of our initial thoughts of what is possible. But we hope, as I mentioned, we begin to take for granted on what it actually takes to do research, again, from a perspective of it's a new paradigm that we all can participate and is no longer novel to advance the mission on behalf of children. So with that, I'll end here and turn over back to Vanessa, but really with a immense sense of gratitude from the Data Resource Center for all across our community and NIH who have made this possible. At every step, we have had to challenge the paradigm of what is possible, and it would not have been able to be challenged in the absence of that collaboration and innovation.
Thank you, Adam. What a wonderful just summation of the science and technology that's really led us here. As you mentioned, it really has been something of a paradigm shift in the last 10 years. I'm excited to talk about that a little more. So I'm so excited to introduce our panelists in our impact across communities and research. So first up, we have Dr. Mary Marazita. She is a distinguished professor of oral and craniofacial sciences and human genetics at the University of Pittsburgh. She is a Kids First investigator who has contributed to multiple Kids First oral facial studies in the portal and the scientific field at large. Dr. Marazita, welcome. I do know you have a...
So thanks for the introduction and welcome, Vanessa. I'm just going to give a couple minutes summary of the trajectory of our studies of oral facial clefts as an example of the other studies in structural birth defects that are part of Gabriella Miller Kids First. So in particular, I'll be talking about our Pittsburgh oral facial cleft studies, which have the major goal of understanding the genetic basis of oral facial clefting, one of the most common congenital anomalies in humans worldwide, occurring in about 1 in 700 births. The lip and palate develop very early in pregnancy, starting about week 4 and finishing about week 11. It requires a lot of very complex and precisely timed growth and fusion of a variety of tissues to create the lip and the palate. If anything happens to disturb that development, a baby could be born with a cleft lip, a cleft palate, or both a cleft lip and cleft palate, with large variability in expression across individual cases. And these cases are seen in every country, every ethnicity, every part of the world. So in the POFC study, we started in the 1990s to collaborate with people around the world to develop genomic and phenotypic resources from diverse populations worldwide. And to date, we've recruited about 23,000 individuals from case families, of whom 9,000 were affected, plus 4,000 pure controls with no known personal or family history of facial birth defects. In the early days, we did a lot of statistical projects. Eventually, there were various types of markers that could be used for studying the genetics, but really there were a couple big turning points for POFC. The approaches that were made available by the Human Genome Project first, and then later on, the ability of participating in the Kids First projects. So Human Genome Project brought common variant approaches to the table initially. They discovered about 10 million single nucleotide polymorphisms, or SNPs, and all of the POFC participants that I showed you on that map have SNP panels through CIDR, another NIH program. And to date, about 90 genomic regions have been identified and replicated as being strongly associated with risk of having an orofacial cleft birth defect. So common variants, SNPs, are great for gene mapping, for finding where in the genome, but most often these are in regulatory regions and are unlikely to be the causal variants, so are unlikely to be able to tell particular individuals or families what is going on in their families. Now, rare variants are more likely to be causal, and although there are certain SNPs that represent rare variants, you are able to identify many more through whole genome sequencing and structural variants. To a certain extent, now using multi-omic approaches that Adam introduced as well. I'll talk a little bit about that in a minute. So that's where I started our involvement with the Gabriella Miller study, and have been very fortunate to be a participant from the first year of funding. We had a project that allowed us to get whole genome sequence for 430 of the children from POFC and their parents. And as this press release said at the time, this was the largest whole genome sequencing effort to examine an oral condition that was initiated by NIH. And we're very proud to be part of that. Now, there have been many more studies in our group and others. But through Kids First in particular, there have been 10 OFC projects funded since it started, and represents whole genome sequencing of almost 3,000 trios, almost 9,000 individuals, and now a couple new studies that bring us into the multi-omic area, with EPIC panels being under process right now, and about 3,500 affected individuals. Of those 10 projects, 7 come from my group and 3 come from other groups that are close friends and collaborators of ours. So thanks to the progress made possible by GMKF, we have a lot of exciting ongoing projects where we're doing functional studies of some of the variants identified in the whole genome sequencing from GMKF in mice using CRISPR-Cas technology with the Jackson Lab, expanded structural variation studies and joint studies across multiple birth defects with Harrison Brand and colleagues at Harvard and Mass General and the Broad. And the really exciting future step that GMKF is making possible is large-scale epigenomics on several thousand of the individuals who have sequencing through GMKF. And these EPIC panels give a reading of methylation sites throughout the genome that will be analyzed by our group and others, led by John Schaefer at Pittsburgh. So thank you, GMKF, for making all of these future programs possible. And as Adam and others pointed out, the resources from the POFC studies are available to other researchers. All of the SNP panels are on dbGaP. The whole genome sequencing from Kids First is available at the Kids First Data Resource Center, and also at dbGaP. And all of the comprehensive phenotype, clinical, imaging, and all the other data on all the families in POFC, all the individuals in POFC, are available through FaceBase. So impossible to thank everybody personally, but thank you from the bottom of my heart to all of our collaborators, and especially all of the participants worldwide.
So next, I'd like to present Dr. David Tichy. He is a professor of pediatrics and holds the Distinguished Endowed Chair in the Department of Pediatrics at the University of Pennsylvania's Perelman School of Medicine. He is a dedicated physician scientist at the Children's Hospital of Philadelphia. Dr. Tichy is a Kids First investigator whose main focus is on acute lymphoblastic leukemia, where he's deeply involved in both clinical care and research. Welcome, Dr. Tichy.
Thank you for having me.
Next, I'd like to present Ellen Miller. Ellen Miller is the president and founder of the Smashing Walnuts Foundation. As many of you know, Ellen is also the mother of Gabriella Miller, after whom the Gabriella Miller Kids First Pediatric Research Program is named. She's a renowned leader and advocate, well known for her work in advancing legislation, policy, and research in behalf of patients and their families. Welcome, Ellen. You have heard from him before, but Dr. Resnick is also on our panel, and he will be providing just a wealth of expertise from his vantage point in the Data Resource Center. I'd like to kick off with just kind of asking questions about really the milestones and the discovery that's been enabled through the Kids First program. So, Dr. Chichi, it's really interesting seeing your work in acute lymphoblastic leukemia. And I was wondering, what is the most surprising shared biological pathway or genetic link that the Kids First data has helped you uncover? And what does that mean for treating these conditions and applying them to the patients?
Thanks. That's a great question. First, I'd like to thank the Gabriella Miller Kids First program and the NIH for all of their support over the years. It's such a fantastic program that's really helped enable the research our team is doing, and is a large, very collaborative effort that my team is doing, and all the other important projects as well. There's no other mechanism that's like this, and you guys are so great to work with. So again, just like to thank you for all the breakthroughs you've helped make happen. As mentioned, my group studies acute lymphoblastic leukemia, which is the most common childhood cancer. This is a cancer that was universally fatal 50 years ago, but nowadays the overwhelming majority of children are cured, but we're not curing everyone, so we still have lots of work to do because that is the goal, to cure every child with both leukemia and any type of cancer. ALL falls into two main types. There's B cell acute lymphoblastic leukemia, and there's T cell acute lymphoblastic leukemia. TALL is about 20% to 25% of cases of ALL, depending on the population of interest. And there's been a lot of breakthroughs over time with BALL, but not as many with TLL. It kind of piggybacked on BALL work and trials over years, but over time they've separated the way we've treated them. And there's just not been the same level of work or understanding of the biology and novel therapies for TLL. But thanks to projects like the Kids First Project, that is changing. I think there's been three areas that BALL has kind of been ahead, that the Gabriella Miller area program has really helped us catch up. One, which is directly related to your question, which is risk stratification. So in patients with leukemia and other cancers as well, when a patient is diagnosed, combining clinical features, demographic features, response to therapy, and very importantly, tumor genomics are used to identify which patients we expect to do very well with standard therapy and which patients we expect not to do as well, and then tailor therapy accordingly. And for those that don't do as well, we do things like either more intensive chemotherapy or take them to stem cell transplant or use new agents or new immunotherapies or targeted therapies right out the gate. And that's really improved outcomes in BALL. And TLL, we've not really been able to do that. And currently in the clinic, the only thing that's used in risk stratification is clinical response to therapy. So we're very good at identifying 3 to 4% of children who do very badly, but everybody else, the other 97% of kids, get treated the same when they first walk in the door. So most children who relapse or it comes back, we think are low risk when they're first diagnosed. And the genomic data we've gotten through the Kids First project have really helped change that. So Gabriella initially funded a project to do whole genome sequencing, whole exome sequencing, and what's called transcriptome sequencing on 1,300 children with TALL, all treated on an NCTN-sponsored NCI clinical trial, ALL 0434. So we could systematically look at a large clinical trial and all the children treated on that trial uniformly across over 220 centers in the US, Canada, Australia, and New Zealand to try to understand more of the biology. And I think the biggest thing that came out of that wasn't necessarily a specific pathway or genetic link, but the importance of non-coding alterations really in TALL biology. So when we think of genetics, most people think of changes in genes. Those are what encode proteins, which are the building blocks of cells. There's also, that's about 1% of our DNA. Most of our DNA is what we used to call it junk DNA. Now we know it's regulatory elements. It's things that turn on and off genes, which makes sense that there's a lot of that's in our DNA because a hair cell and a skin cell and a blood cell are very different, but at a DNA level, they all look kind of the same. So why does a hair cell turn into a hair cell? And what we found in T cell leukemia, 60% of the genetic changes that are leading to leukemia, Where the changes are is actually not in the gene itself that makes the protein. It's actually in something that turns on or off a gene. And prior to this large Gabriella-Miller's effort, all of the sequencing that had ever been really done at any scale in TALL was looking just at the genes themselves, looking at exon type sequencing. And since 60% of what actually matters isn't in those parts of the genes, that's why we've never actually really been able to identify the changes in the genetics that actually make a difference, that are actually leading to leukemia. And this has allowed us now to understand more about why children are getting leukemia, but also for the first time to really be able to risk stratify patients. So now we can identify those children when they're diagnosed that are going to do not as well, so we can start talking about different therapies. But also, just as importantly, which kids are going to have a 99 to 100% chance of being cured walked in the door, where we know we're giving them too much therapy. Our chemotherapy is very toxic, so it's letting us starting to ask questions like take a step back. Um, the Kids First program has also funded a large project to sequence the successor trial to 0434, which is the ALL 1231 trial, just so before we start changing how we treat children with leukemia across the US and the globe, we want to validate those results and make sure that everything actually holds up, and the genes that we found in one study actually holds in another study. And I think there's a lot of downstream consequences as far as precision medicines and immunotherapies we can talk about more, but I'll give some time for the other panelists to answer questions.
That was a robust answer. Thank you so much, Dr. Tichy. Some of your insights kind of lend into another question I actually had for Dr. Marzita. We kind of we've touched on this kind of as a through line throughout all of your presentations about kind of the tools and the sequencing types. Dr. Marzita, I know that you mentioned some tools and sequencing types that POFC has been able to leverage due to the Gabriella Miller Kids First program. Could you share an example of how that ability to access and analyze large-scale genomic and clinical data in the Kids First Data Resource Center portal has enabled a discovery that might have not been possible using other research models? I believe you.
Sorry, I'm not on top of my not on top of my mouse today. So the whole genome sequencing has been a big game changer for the types of questions we're able to ask and hopefully eventually answer with birth defects like oral facial clefting. So the one thing I did not mention in my initial presentation was that we are studying isolated oral facial clefting, and that is individuals who have the cleft defect but no other major physical anomalies. There's also cases that are said to be syndromic, where they have multiple types of birth defects. And there now are clinical genetic resources that involve sequencing panels that are used with syndromic cases. And they cover maybe 200 genes, the 200 major genes that are important in the syndromic cases. So when a child is born with multiple birth defects, including the face, you can run this clinical panel and identify the gene that's important in your child. So one of the students working on our projects in the lab of Elizabeth Leslie Clarkson at Emory University took a look at the genes that are sequenced in those panels within our isolated cleft cases. And in about 10% of those isolated cleft cases, there were concomitants within those syndromic genes, highlighting the possible importance of those clinical panels for isolated cases as well as syndromic cases. So I think that's very exciting because one of the important next steps for us is to identify causal genes. You know, a lot of the things we've seen in the SNP projects, for example, as David alluded to, are not the genes that actually cause the defects. They're in the regulatory regions. So if we can find genes like that panel, if we can find variants like in that panel, we might be able to get more specific information to families. So it's exciting to see that idea has merit from our GNKF sequencing results.
A great insight. And I actually had a follow-up question for you. So I know just your span of and your tenure with all your research, I was curious about kind of the landscape you've noticed. And Dr. Resnick kind of touched on this, the different tools available, starting from, I think you said 1998 was the POFC study. And from that time to now, I'm really curious about how those tools have changed and how that's really lent itself to your work.
Well, one of the really telling points about how things have changed is that I had a grad student who was reading some of our older papers, and he came up to me after he'd read it and said, Dr. Marizzi had a really interesting paper, but why did you only use 40 markers? And because now we have millions that we can use. So I explained, we had microsatellite markers, we had this and that, but we did not have the tools and facilities that are available to current researchers. So it's been really exciting to go from, 40 markers to 40 million if we want. And programs like Gabriella Miller, the CIDR program that was a precursor to get us SNP panels, it's just allowed a snowballing of the tools available to researchers. So it's super exciting. And I know it's going to lead to a lot of very important discoveries over the next few years. Just really amazing.
I'm amazed too. I mean, 40 million, did you say, or 4 million?
Well, if you had enough money, yeah, you could get 40 million.
Well, I couldn't count that by hand. That's amazing. I did want to ask another question to Dr. Tucci. So this is, you did mention a little bit about your clinical trials, the leukemia clinical trials that you've led. And I was wondering, how has the comprehensive genomic profiling data available through Kids First translated into new therapies or precision medicine approaches for children in your care?
It's a great question, and it's a big problem. And I think the Kids First program and the data that's being generated are really helping that a lot. I mean, in TALL and lots of childhood cancers, there's lots of exciting new agents, precision medicines that are out there. The problem is like when we've been non-specifically taking those drugs and using them in a petri dish against different cases of TALL. There's no one drug that works in everybody. Most drugs work in about 30 to 40% of cases. And we've never really been able to identify the patients in the clinic that are more likely to benefit from the drugs. Unlike some cancers where it's very easy, match a mutation in a certain gene to that drug and you expect it to happen. As I mentioned in TLL, the biology is a lot more complex. A lot of the genetic changes are in the non-coding parts of the genome. But they do turn on certain pathways. And I think we've now can identify in certain genetic types of TLL which patients are more or less likely to respond to different agents. And we already have clinical trials that are open in the US and clinical trials that are open in Europe that are taking the Kids First data and asking those questions. Could a patient with this genetic change benefit from this particular drug? I know of individual patients who this has actually happened in the clinic already, like children who have had TLL that was very refractory to therapy. We did genetic testing. We then looked at the Kids First data that we had, which could kind of predict they might respond to this drug. And sometimes we then use those drugs in the clinic in more of a compassionate access. And there's children who have actually benefited for that. Of course, the goal is not to do these non-clinical trial to learn if things work. It's actually to build and run large clinical trials so we can actually definitively answer those kind of questions. But this has allowed us to do that. I just got back from the American Society of Hematology annual meeting that was in Orlando over the weekend. And it was really nice to see that, and people have commented on this afterwards. There was a bunch of talks on TALL there this year. And every single one of them started off with actually showing the TALL genomic map that we actually made because of the Kids First data and how it actually impacted their research, whether that be biology or a lot of it was therapeutically driven, where they basically said, because of this, we were studying this drug, and then we tied that together, and these are the patients that we were going to see that benefit. And that was a theme that had over and over and over and over, which I think is the most powerful thing about the Kids First program is it's not what does it just do for my lab and the stuff that I do, but it's actually a resource that so many people in the community can use to help benefit their patients as well, and tailor to the patients they're treating, not just in the US, but across the globe.
David, I wonder if, Vanessa, if you don't mind, the other thing that you have really championed, and particularly in TLL, is given the fact that some of these genomic alterations occur in non-coding frameworks, you've also leveraged the Kids First program as some of the pilots that really demonstrated the value proposition of bringing proteomic data alongside. I wonder if we can make some comments on that front as well.
That's a great point. Two things. One on that is the proteome data are very important because since we are targeting non-coding things that lead to upregulation of different genes or the expression of those or downstream things, ultimately those change proteins. And we have theoretical expectations of which proteins they're actually changing and which they're activating. But the proteomics of the inside of the cell is actually allowing us to definitively answer that so we can actually make sure that our predictions more hold up in biologic truth. But also importantly to that is for immunotherapy. So one of the differences I said between B cell ALL and TLL is in BALL, immunotherapies have really transformed the therapeutic landscape. Things like bispecific T cell engagers like blinatumomab have been shown in randomized trials to improve and cure more kids. There's CAR T cells that are on the news and people talk about all the time. TLL, we really haven't had immunotherapies make the same impact. And part of that is because if you immunotherapy basically attacks something on the outside of a cell to let the immune system work like it's supposed to do, the part of our body that fights an infection. The problem is T cell leukemia arises from normal T cells. And we need our normal T cells. Our normal T cells are what fights viruses and funguses and lots of other infections. And if you wipe out your normal T cells, you basically give somebody HIV. So an immunotherapy that wipes out both T cell bad cancer cells and normal T cells is basically changing cancer for AIDS. That is not a win. But what the Kids First data is letting us do through proteomics is actually to compare normal T cells with T-ALL blasts at a large scale to see what proteins are different so we can develop and identify and build new immunotherapies that we can move to the clinic. We've already identified some exciting targets, and we're already starting to build some new immunotherapies and CARs in the lab that hopefully will translate and be in patients pretty quickly. It's an exciting time in science because you can make those discoveries and then actually translate them into a new therapy and get it to the clinic in my lifetime, where it used to be that kind of thing would take 30 to 40 years before it actually would impact a patient versus a couple short years. So other work that would never have happened without Gabriella Miller.
That was a great question, Dr. Resnick. So Ellen, I'd actually like to ask you, just as the mother of Gabriella Miller and a renowned advocate, I would really like to hear your thoughts after hearing what the impact on the researchers, and really what does that kind of translational science and what do those milestones mean to patient families and advocates?
Well, this all started because a little girl told our elected officials to stop talking and start doing, but she wasn't quite so polite as that. She used a bad word and said talk is BS. We need actions. And so her sassiness caught the attention of our elected officials, and they had a piece of legislation that was not moving. And I've had multiple elected officials that were working, even those that weren't the originals working on that piece of legislation, saying that without Gabriella, it never would have passed. So that is humbling in and of itself. Keep in mind, Gabriella had DIPG, diffuse intrinsic pontine glioma. This is the same cancer that killed Neil Armstrong's daughter back in 1962. Gabriella was diagnosed 60 years later. It was the same protocol 60 years later. It was a clinical trial and radiation, and it was the same outcome, 6 to 9 months. And so from that, a couple of years after Gabriella died, with this program starting at zero, Dr. Resnick had said earlier that now specifically for DIPG and DMG, they've got over 12,600 bits of data research that they've done specifically on that, going from zero. That's more than they did in over 60 years from the time that Neil Armstrong's daughter was diagnosed. Dr. Resnick also said that we've got over 1.56 million total data files. starting from zero, over 200 peer reviews. Dr. Brody said that there are over 30,000 patients and families now that we've got whole genome sequencing, and over 1,000 approved data access requests for secondary users because of the data that this program has found. Dr. Tichy had just said that the data is being used globally, and it's across different diseases that share common mutations. So this came from nothing. And in 10 short years, wow, we've done incredible research. So hats off to all of these researchers across the country and around the world that are doing this work.
Well, being one of those researchers, I think all of us also not only grateful to Gabriella herself and the chain of events that she initiated that resulted in the past 10 years, but for us, it's also of immense importance to recognize, I think, as every one of us say, that there's more work to do. And the fact that you have continued to be a champion of the program in ways that now set the stage for the Kids First 2.0 Act. I wonder if maybe you can just say a few words on that as well.
Yes. So one thing that I'll say before I jump into that is, again, hats off to Gabriella. I've had countless researchers say that now they have faces to go with the research. So they're in their labs and they're working on a specimen, but the specimen was a child. And now there are countless faces that are attached to the research that they do. And it's because of Gabriella and every other child that has lost their life to a disease and has donated post-mortem that they've humanized. And that's amazing. And I love that these researchers have a new drive towards finding cures for our children because of that. Okay, so yeah, I'm sorry, Adam, you were saying.
I was just one expressing my gratitude, but the fact that it's thanks to your continued advocacy that we now have Kids First 2.0. And I wonder if maybe you could just say a few words about Kids First 2.0.
Yes. Okay, so the original piece of legislation was just a phenomenal success, as we all know. And we all knew that it couldn't stop after 10 short years. We don't have cures for everything at that point. And so what was fantastic is this program and countless other pieces of childhood cancer and disease legislation have led to the patient advocates bonding together to really become an incredibly strong voice, to meet with our elected officials and get to a place where until we have what we want, we're not stopping. So that's actually what's happened. I mean, hats off to our patient advocates across the country and around the world doing this work, because we could go back to the time of Neil Armstrong. That family was told, your child has terminal brain cancer. There's nothing we could do. Go home and make memories. We're not okay with that today. And we've got access to information and to people worldwide, and we are sharing information. We are reaching out to our elected officials. We're reaching out to our researchers. We're tearing down silos and barricades and saying, nope, that's not good for us. We know that we could do more. And look at the results. I mean, look at these folks that are on the panel. They are blowing things up with giving new information to getting cures. And we certainly, as the advocates out there, are not going to stop. We need the 2.0s out there, and we need to make certain that it goes on beyond that, the 3.0s and so forth, to ensure that you never have a shortage of funding.
Ellen, we thank you for your advocacy. So I did have a follow-up question what do you believe is the most critical area of program support, advocacy, and community engagement that could really that you believe that should be championed to really ensure the program's long-term success?
Well, number one, as a patient advocate and speaking on behalf of all the patient advocates here in the United States, we need to make certain that our voices are heard by our elected officials. We need to, it doesn't matter what side of the aisle they're on. It doesn't matter whether or not we voted for them. We need them to be our friends and our advocates. And what's pretty amazing is that we're doing that. So not only for this Kids First, but all pieces of legislation that can help childhood cancer and childhood disease, that I think is one of the most critically important things that can be done. And what's great about that is that there's no shortage of that. I'm actually on a Zoom call later this afternoon with a senator to talk about another piece of legislation that we need to get passed. So we in the advocacy world band together to ensure that we get this passed.
And maybe just to give additional credit to where I think you often are a little too humble, Alan. I think that what has really been amazing for us is that as we have been advocating for greater integration across a collaborative framework that brings together different cancers in a common research setting, but then expanding it to beyond cancer to, for example, craniofacial disorders or conditions. You have mirrored that on the advocacy side, and you yourself, even with a focus on DIPG, have expanded to partnering with other foundations and families and other cancers or congenital birth defects. And I think it's really that synergy of both community partnership and research partnership that's really a special and very different model of working together. So we on the research side really appreciate what you have done, not just on advocacy with legal actions at the government layer, but also at the foundation layer, bringing folks together. The number of emails and texts I get from you that so-and-so in this place has this, they want to talk to you about Kids First. Like your advocacy and connection, I think is really terrific. And again, very novel and different than the way most foundations have had to operate historically. So I just want to say thank you for those efforts.
Thank you. Thank you. Many people in our DIPG world and other worlds where there's a disease that has no or horrible cure rates always say, what if? I mean, this is not a new disease. What if the Armstrong family had access to other families that had this same disease and they banded together? How much further could we have been along with finding cures? And so here we are. And honestly, for every single family out there that is just bereaved, heartbroken, tired, it is certainly okay for them to say, I'm done with this. Like, I need to live my life. And there's nothing, nobody thinks badly of that. But then these folks see that there are people that offer support. They've walked in their shoes. They get that. And we band together and we form this amazing, tight-knit group and realize that we have a very strong voice together. And what's very exciting is exactly what you've just all said. In the past 10 years, what has happened? We've done more in the past 10 years than had been done in the past 70. So it's exciting times. And we see that as the advocates. And now the question is, what if? what if we could get more funding? What if we could get in front of more elected officials, get more legislation passed? What if we could get more money into those that are doing the research? How much further could we get? And it's exciting times for all of us.
I want to jump off that momentum, as you mentioned, Ellen. It's really wonderful to hear your perspective. Dr. Resnick, as the scientific lead at the Kids First Data Resource Center, what new features or advancements on the platform are really going to kind of drive that momentum like we've been hearing from all week or all morning?
Oh, yeah. Actually, I was just answering one of the questions in the webinar, the chat, that relates to that. And, really the initial mandate of Kids First focused on covering this tremendous gap of genomic data information as paired with an initial phenotype, like a certain cancer condition. But the next phase is really further enriching that, both across modalities. So like I mentioned, expanding long-read sequencing, a new technology for the genomic and transcriptomic analysis, or proteomic analysis, like David mentioned. But paired with that is really even more challenging a domain, but one that we're making headway, and I think Kids First has an opportunity to further advance, is what is the deeper and even more diverse clinical data elements for these patients. So each one of these patients has longitudinal clinical outcomes, both as part of clinical trials like the COG trials that David mentioned, but also data that lives within electronic health record systems and clinics. And that's actually one of the most challenging activities that many of us undertake, is the integration of that clinical data across time and linking that to the genomic and molecular features. And I think some of the investments that the Kids First program has made in a specific type of architecture and data workflows, including the adoption of fast healthcare interoperability resources, or what's called FHIR, in partnership with the Office of Data Science at NIH and other partners, have really set the stage for us to begin experimenting with how to bring more of that data and integrate it across time. So in many ways, the data is not static, in the view of the portal for particular patients, because we continuously add data across time to that patient in ways that drives additional analysis and interpretations across patients, not just those patients within the portal, but across patients within the portal and those that are coming to clinic. David sort of hinted at that potential, as part of the diagnostic and decision-making process. So that process of more near real-time integration and use of the data that builds on top of foundational discoveries of a causative gene or mutation is really the key next step to drive transformative care. To achieve that, we need to also build new and adopt new technology frameworks. And I think this is one area of tremendous excitement for us and many others. In fact, I'm actually here in Boston meeting with a team advancing some of these efforts as part of several other federal initiatives that are focused on the adoption and use of responsible artificial intelligence, or AI. How to bring those technologies in ways that augment and support the research endeavors, accelerate that process in ways that are still really at the discovery leading edge. But what AI does need and want is a lot of data. So the more data you have, the better AI performs. And that's really where the opportunity landscape is beginning to emerge in the context of the Kids First Data Resource. Now, these are still new technologies. Even we as humankind are just coming to terms with the use and implementation of AI in our daily life beyond just a ChatGPT framework. But in many ways, the part that is exciting for me is the opportunity for the pediatric and rare disease context to be at the leading edge of those proving grounds. Historically, it's at times been a narrative that new technology get developed and optimized in the setting of adult or common diseases, and then percolate down into the rare or pediatric setting. And that is not has not always benefited our community in ways that harnessed the opportunity landscape and accelerated processes. I think the kind of work that the Kids First program has done has demonstrated true innovation of what's possible and how to do science in new ways. That also has set the stage for how it can become a proving ground, a responsible proving ground for new technologies like AI. And we'll need to recruit and bring additional researchers into this domain, AI researchers and others. But it represents a new frontier that we are eager to really advance and test on behalf of the pediatric setting.
What great insights. And I really it's so just amazing to see how data interoperability and just that strong foundation of the program has led to translational sciences and hopefully future integrations into electronic health records. So I did want to ask one last question to all of our panelists. Looking ahead at the next phase of Kids First, what is one major goal, whether it be a specific clinical advancement, a research discovery, or a community benchmark that each of you hopes to see achieved in the future?
Who do you want to start?
Go ahead, David.
I mean, it's probably not what you were expecting me to say. I'd like to see the big thing, and I think a focus, and I don't know the best answer on how to do this, is educating and informing the community at large on the importance of what you are all doing and what we're all doing. As a physician and a scientist, I've like spent my whole career going around and talking to doctors and scientists on what we do and trying to explain how important it is. And as was just told right now, all the great work that's done through advocacy, going to the government and working with politicians to inform them how important this is to get the funding to continue the work. What I didn't realize until recently is that as a gap is the importance of making sure the community at large realizes the value that this kind of work adds to society, to their families, to their friends, to their patients in general, to make sure the messaging that science and medicine are a good thing, and we need to continue to advocate for them and pushing the needle forward, and how that ends up helping everybody and benefiting everybody through social media and other aspects as well. So I think could be figuring out a way to engage the community at their level so they just see what we're doing in a better and more transparent way so they understand. Because they're the ones who pick the politicians that we're trying to convince to give us more money.
Yeah, I agree. That's really vitally important. And the community of families has become much more knowledgeable than they were like when I was starting out. So, people just simply would blame the mother if a child was born with a birth defect, but we know it's more complex than that. And the general population realizes that now too. So we're getting much more probing questions about where we're going with our research. So that ties in exactly with what you're talking about, David. And that is a big frontier in general. And ways that Kids First can contribute to that are exactly what is happening with the addition of multi-omic modalities that can be requested, such as the epigenome, are going to be really important for birth defects and for cancers as well. The crosstalk between cancer risk factors and birth defect risk factors needs additional exploration. We're already finding, lots of commonalities, and that's a beauty of the way Kids First has been designed, I think. And finally, just being a beacon for pediatric research. You find a lot of money for heart disease. We need to keep finding money for our children as well.
Yeah, I think that both what David and Mary said is great insight, and which at least I fully align with. And from a data resource center perspective, for me, what drives us, across our partners is really to continue to innovate on the how of doing science in ways that intersects with the kind of things that David and Mary, I think, championed a second ago. And that how for us really is about speed. Can we accelerate how research is done, how discoveries are made? Time for us is the factor of optimization. And so our goal is really to find new ways to drive that accelerated discovery process in ways that we can continue to optimize. It's clear, even just across the history of what we've been doing, that we can accelerate research through these efforts and accelerate discoveries. But we also believe that there's even more we can do to drive that process through the kind of partnerships and ecosystem we've been developing. So that for me is a key milestone, and we want to continue advancing.
For me, as patient advocate, very simple. Just keep advocating and ensure that the funding doesn't ever go away.
It was great hearing from all of you today. I just want to say thank you to the attendees for attending, and of course our panelists for their amazing insights and participation. A key takeaway for me personally is just the immense network of just interdisciplinary knowledge and talent that we have across the program. It's really been a pleasure just moderating this conversation today. And I just like to say one big round of applause for all of our panelists and speakers. Thank you so much, and I hope you all have an amazing rest of your day.